Good morning, all of you. I warmly welcome you all for the first lecture of the Distinguished Lecture Series conducted by the Open University of Sri Lanka, especially by the Center for Education, Technology and Media in collaboration with International Unit and also Information Technology Unit of the Open University of Sri Lanka. So this is the first lecture of the fourth series we started a couple of years back. Today our distinguished guest speaker, Dr. Janaki Lelwala Guruge, Senior Research Scientist, Center for Genome Sciences and Systems Biology, Washington University School of Medicine, St. Louis, Missouri, USA. We warmly welcome Dr. Janaki Lelwala Guruge as our distinguished lecturer for the first lecture of the series in 2018. We welcome the Dean of the Faculty of Natural Sciences, Professor Leelani Senaratne, and also the Dean of the Faculty of Health Sciences, Professor Gaya Ranawaka, and all the distinguished members of the Open University, academic and administrative staff. This lecture series we stream lively, so anyone can watch from anywhere of the world, and also simultaneously we record the lecture and upload to Open University of Sri Lanka website. So later also you can refer to this lecture. Dr. Janaki Lelwala Guruge graduated, first completed the tertiary education in Visakha Vidyalaya, Colombo, and joined the Sri Jayawardhanapura University for her bachelor's degree. She got bachelor's biology honors degree and master's degree MPhil also from Sri Jayawardhanapura University. She then proceeded to Lund University in Sweden, where she successfully defended her doctoral thesis working on antigenic properties of Helicobacter pylori. Although this bacterium is now considered as a carcinogen by the WHO for its pathogenic properties causing gastritis, ulcers leading to cancer. In 1994, she joined the School of Medicine at Washington University School of Medicine in St. Louis for her postdoctoral studies. Currently, she is a senior scientist with Dr. Jeff Gordon, a world-renowned scientist leading research on the human microbiome. Her expertise includes techniques to create arrayed libraries of cultured bacterial isolates of human. We are looking forward to her talk on current techniques used to understand the microbiome, the future of medicine, and also we hope that she will also discuss about the impact of microbiome on human health. According to my knowledge, she also involved in Human Genome Project second phase, and after sequencing the whole genome of human being, now we are looking into the microbiome, genome of the microbiome. We know that there's a strong impact of microbiome on human health. And we welcome her husband, Bandula Lelwala Guruge, as well, all the way from Washington to Sri Lanka. And I hope that this audience is not an alien's audience for her. She has a lot of schoolmates, classmates, batchmates, and I warmly welcome Dr. Janaki Lelwala Guruge to address the audience.
Thank you very much for that kind introduction. I've known Jayantha for a long, long time. We are both products of Sri Jayawardhanapura University. So I think if I do well today, we can share the credit. And if I don't do well, it's all on you for inviting me. I'll see what I can do. So I will give you a brief introduction, and I will explain the techniques that we do to make arrayed culture collections of human donors. And if you need any further details, please don't hesitate to contact me through Jayantha. He has all my information. So just before leaving for my vacation, I met a physician, and when he heard that I'm working on the microbiome, he said, wow, that's the future of medicine. So if it is that important, let's see what the microbiome means. So the word microbiome is used to explain a collective genomes of the microorganisms that reside in any given environment. The word microbiota means the community of bacteria, archaea, fungi, viruses, and protozoans that reside in the same community. But for the purpose of the talk today, when I use the words microbiome or microbiota, I'm only referring to bacteria, because that's what I'm working on. And I'll be talking to you about the microbiota of the gastrointestinal tract, because that has become very important in health and disease of humans. So how did we get to this point to talk about all these microbes? We know that the screen is off, I think. Oh, you didn't see the first slide even? First slide? First slide even? Okay. So the Earth actually began billions of years ago. And according to this, the first life, that's microbes, arrived several billion years before the humans. When humans arrived, the Earth was actually colonized by microbes. They covered every surface of the entire Earth. And we, the humans, arrived, of course, uninvited. And bacteria found new niches, and they colonized humans inside and outside. Wherever they could live, they started colonizing. And for a long, long time, over millions of years, microbes and humans co-evolved, meaning they were like beginning to understand the environment, and they were helping one another. This is just to give you an overview of how much microbial we are. So it is estimated that less than a third are human cells in our body. The rest of the 90% are bacteria, fungi, and protozoans. And the same goes with the genes, because each bacterium, each fungus carry their own genome. So we are more microbial than human. These numbers keep changing, because scientists find new ways to calculate the numbers. So I think today it is thought that we are 50% microbial and 50% human cells. So even a 50% is a lot of cells. So why do we carry so many microbial cells? Obviously, that must be very important. We all know that babies are born sterile. They acquire their first microbes from the mother when they pass through the birth canal. So obviously, there are different communities during vaginal delivery and C-section. As of today, I don't think we can pinpoint what bacteria are important in health, but we all know that they have different communities. Same goes with breast milk and formula-fed babies. So breast milk-fed babies carry more bifidobacteria, and formula-fed babies carry different bacteria. These communities have been very extensively studied. And I can tell you a little story here. So the mother's milk carry a very important oligosaccharide, which is called a silylated oligosaccharide that is only digested by a bacterium named Bifidobacterium longum infantis. So the question is, why would a mother provide an oligosaccharide for a bacterium when the baby cannot digest it? So those are the questions that we are trying to answer. So more bacteria acquired from the environment and from family members as the baby grows. The bacterial populations that are acquired at the beginning, early in life, continue to be the same, but there are shifts in populations because of the diet that we take. Because the gut is a very subtle environment for microbes, because we keep changing our diet and they get flushed with all this diet, and they have to hang on there and learn to live in this changing environment. We also know that the host, so how does the host distinguish between the pathogenic bacteria and the good bacteria? We've been able to show in a mouse model that there is a very low antigen response to the good bacteria, so that the host can keep them in balance. And the host is able to actually keep these 50% of microbial cells in a human body without mounting an immune response and recognizing them as pathogens. I think that's an amazing thing. So today we know that the microbiome is blamed for everything, starting from acne, diarrhea, asthma, allergies, autism, autoimmune diseases, cancer, depression, anxiety, you name it, it's related to the microbiome. Then we also know that the microbiome helps digest dietary fiber. There are a lot of activities that the human host cannot do that the microbes have taken over. That's because we have been co-evolving with the microbes for over millions of years, as I just mentioned. One of the very important things is that you may already know that humans cannot synthesize vitamin K. It's a bacterium that helps synthesize the vitamin K that we really need. Then there are also the short chain fatty acids that is very important for colon health that the humans cannot make. Only the bacteria in a microbiome can do this. So in other words, the bacteria help us maintain normal functions of the gut. However, there are rapid shifts in the microbial population too, because of the different diets that we take in. These shifts in mice can be measured within several hours, even over in one day. So this actually tells us that the diets should be very, very important to maintain a healthy bacterial population. We also know that subtle imbalances in our microbial populations can cause disease. One way that we destroy a healthy microbiome is taking the overuse of antibiotics, as we all know, because a lot of people take antibiotics even for a common cold. And I think that should not be done, because people don't realize that it kills the pathogens as well as the good bacteria. And people have seen that restoring the balance would lead to cure. But all these studies are still underway, and someday we'll get there. I like to switch gears here to tell you a little bit about my favorite organism that I worked on for several years, the Helicobacter pylori. Since it causes gastritis, ulcers that lead to cancer, the World Health Organization at some point declared it a class 1 carcinogen. And it advised every physician to treat the bacterium with antibiotics, which we have done. And today I think the Western population, this generation, is the first generation that's born without carrying Helicobacter pylori. This bacterium was found in every human being, and its absence is correlated with upper gastric disease. Although it causes gastritis, ulcers, and cancers in the stomach, people are now beginning to realize that in its absence, the esophageal diseases are rising. H. pylori has co-evolved with mammals over 100 millions of years. Recently, there was a paper describing the DNA that they isolated from a mummified human being that was several million years old. So the question is, at what cost that we would like to remove this bacterium from the human population? Because people have seen that even the bacterium migrated with us out of Africa over millions of years, and everybody was carrying the bacterium. At some point, maybe it got a pathogenic gene or something, and some of the strains became pathogenic. But that is also because of, maybe because of overuse of antibiotics or something. We don't really know. So how do we study the microbiome? People use the terms microbiome and microbiota very loosely. One microbiota, so I will be doing the same thing here. Don't worry, it's all about the bacterium and the microbes and the genes. We've been able to show that a fecal sample represents the entire microbiome in the gastrointestinal tract. And there are people commonly use 16S ribosomal sequencing to identify bacteria in a community. I'm not going into details of how we do the 16S sequencing, but if anybody is interested, I can let me know, send me an email, and I can send you the protocols. So sequencing, by sequencing a whole sample, we can identify what bacterial species are in there. We can even calculate the abundance of each species. But what we cannot do is to distinguish between dead and live bacteria, because the PCR would amplify dead DNA, dead bacteria rather, DNA. And then these samples also contain DNA that we got from food, because some food are already contaminated when we consume. So there will be some bacterial DNA. So because of that, we have to culture these bacteria. For in vitro studies, this is somewhat a common technique that people use, which we don't use in the lab. But you can set up something like this. It's called a simulator of the human microbial ecosystem. So each beaker or the container consists of a different pH, and then it simulates the entire GI tract, and you can introduce a bacterial sample and take samples from each container and see what communities, how they build communities, and what communities that you can isolate. The only thing lacking here is the immune system. So therefore, our lab prefers using germ-free mice. I'll tell you a little bit about maintaining these mice. These are germ-free mice because they don't carry a single bacterium. And this gives us a chance to introduce any bacterium we need and understand what its activities, its physiology. Or we can introduce bacterial communities. As long as we know what the input, we can take a sample and analyze the output and see what got into the mice and the phenotype it produces. Raising germ-free mice is a lot of work because they all live in sterile isolators, breathing sterile air, eating sterile food, and drinking sterile water. Actually, they are doing much better than human beings sometimes because they get the cleanest water, cleanest food, and they breathe clean air. We take fecal samples, do swipes once a week, and test them by culture and PCR amplification to see if there are any bacteria. So as long as the cultures come negative, we consider them germ-free, meaning they don't carry any bacteria outside or inside their gut. Then for culturing is also, we have to use anaerobic techniques. It's not like that you can walk into the lab in the morning and say, okay, I need some E. coli cells, start a culture, in 8 hours you get your cells. It doesn't work like that. So we have several of these anaerobic chambers. These anaerobic chambers are equipped with gas tanks, and then we maintain an environment of 5% hydrogen, 75% nitrogen, and the rest is carbon dioxide. Each chamber is equipped with a catalyst that would start eating the oxygen, because every time we bring in something, we bring in some oxygen. Even a single tube has to be, the caps have to be loosened to be able to make them anaerobic. Every culture medium we have to put into the chamber several days before we start culturing, because the medium has to become anaerobic. We pour them outside, they are aerobic, take them inside, and they have to become anaerobic. We use rhesus urine as an indicator for oxygen in the media. So once the color changes, we know that the oxygen has been removed, but we still don't know if that's sensitive enough. So we keep them for like 3-4 days in the chamber before using them. So it's a lot of preparation work. More than the culturing and taking the samples in, we have to have everything, almost everything in the chamber several days before, sometimes for weeks, because there's no way to check if the plastics have become anaerobic. Only the way that you will know that they are aerobic is once you start working, and if you put the medium into a plastic container, the rhesus urine will change color, and you cannot do anything then. So most of the bacteria in the microbiota are anaerobic and microaerophilic. Culturing microaerophilic bacteria is not that difficult, because we can take it outside and then put them into an incubator, a regular tissue culture incubator. The anaerobic bacteria is the technique that we think we have mastered a little bit maybe. The chamber is always maintained at 3% hydrogen. So there are sensors inside the chamber that will tell us if any oxygen has got into the chamber. And then we have to deflate them, inflate them with the gas mix, and turn them back on and start before we start using them. So like I mentioned before, we have already sequenced the fecal sample. And the fecal sample, we know what bacterial species are in there and their abundance. So just by looking at these sequences and knowing what bacteria are there, we can start making media according to, you have to take that out. Yeah. Did I press something? Oh, okay. We cannot recover, unfortunately we cannot recover all the bacteria, but we use several different rich media, a few media that are supplemented with different nutrients according to what we already know, and we also used selective plates with different antibiotics to remove some of the different antibiotic sensitive bacteria. The problem here is the abundance of each species. So some of the bacteria that are most abundant will overgrow, and we will not be able to catch the small amounts of whatever the other bacteria are. So this is always a problem. So because of that, we like to make several stocks of the sample. Once we get one sample, we like to copy it into several different tubes so that we have several samples of the same composition in the freezer. In case we lose something, we can always go back to a second sample and consider it as good as the first sample. And especially for reproducibility, we need to do that. So in this slide, I have put together it's not doing anything. It says it's not currently connected to the internet. Maybe I press that. Can't get the next slide. Oh, that's my most important slide. think I got it, yeah. I think this is my most important slide, although I have it in, I have everything that I need to make an arrayed culture library of a donor. So like I said, we prepare the anaerobic chamber, and then we bring in the sample. This sample is in one tube, and we use like about 0.5 grams to a gram. We don't even need a gram to make a culture collection of a human donor. Once we take it in, we have a step which we call the clarifying step. That point, what we do is make a suitable dilution, mix it with glycerol stocks, and put them into these crimp vials. And these crimp vials are, the stoppers are made in butyl rubber, so that we can take a syringe and a needle and punch it through and take the sample out whenever we want. So these tubes go into the freezer, and each tube should have the same composition. What we have noticed over time is once we freeze thaw, certain bacterial species can get lost. So when we make the clarified sample, we would like to, if we are doing mouse experiments, we would like to colonize the mouse the same day. Because sometimes what we cannot get in vitro, the mouse decides that it wants it and it grows in the mouse, and we can isolate it from the mouse cecal samples. So that's also one way of getting a bacterium that we have failed to isolate in our initial attempt. So like I mentioned, we make several different media, and 2 or 3 days later, the plates will look something like this. Colonies are picked manually, because each student has to make sure that we get everything we want, because this is the most important step in making a library. Once we get this, that's what we have, because after several days, they kind of go into stationary phase and to in death phase, and they die off. One of the problems we have here is this is like treating an entire human population with the same kind of food, asking them to go to sleep at the same time and wake up at the same time. So we don't know, some of these bacteria start coming 2 or 3 days later, but then by then the early arrivals are dying. So we have to sort of pick a time to make sure that we can at least get, we may not get 100%, but at least close to. So then the colonies are picked into a 96 well plate, that each well can, that deep well plate over there, each well can hold 1 milliliter of broth. So now we have, now the bacteria that were on that plate are in a 96 well, deep well plate. So these are very difficult to handle manually. For that we have a liquid dispensing robot inside the chamber. This robot helps us fill the plates. Any liquid transfers are done by the robot. And then we pick these bacteria into the plate and put them back in the incubator and grow them for another 2 or 3 days. Once we are satisfied with the turbidity, we take them out of the incubator and then make copies. So these copies are now frozen in -80, and then we take the remaining bacterial cultures outside and try to extract DNA to do sequence and identify each well. So to do that, we have even a fancier robot. This robot can actually take 96 tip box at the same time. The other one is small, it only has like 8 wells, and so it keeps filling, it takes time. This one can do in one time. And this one is outside, so by then it doesn't matter that the bacteria are dead, right? All we care is about the DNA that we can get out of it. So once we extract the DNA, we do sequencing in the labs. So this time we don't go for 16S, is a 1300 base pairs, which we cannot sequence in the lab. But we try to sequence a 350 base pair region that's called the V4 region. That actually I didn't go into details of the 16S. You can ask me or you can read about it. The 16S RNA sequence has several variable genes that you can distinguish as between different species. So we aim for a shorter region shorter base pair region, and that we can do in the lab. So then we get to see the first shot of our library. Sometimes it's a failure, so we have to face the reality then. Sometimes it has a very good diversity and we are all happy and then we can continue. But for some reason, if we haven't got what we wanted, because we already know what the sample has, right? We can stop there. We can throw out everything that we froze, or we can keep them. And we can go back to a second tube that we froze that has the same community, start again. Maybe with different media, or we have to do something at that point to make sure to get the bacteria out. So by V4 sequencing, we can also see if each well, if the purity of each well, because it will give us different sequences and IDs for each well if it carries more than one bacterium. Because as we all know that even if you do single colonies, each colony could be one single bacterium or it could be several bacteria growing together, which we don't know at the time we pick samples. So after this, we have to purify the sample. So if we are satisfied with the library, we can purify each well. To do that, we have to go back, put them back on plates, streak for single colonies, and pick them back onto a same plate again. Grow them up, do the same thing again, sequence them, freeze them, and then if we are satisfied, at this point, we send the DNA out to do the entire 16S sequence. So sometimes the full-length sequencing does a better job in identifying the species rather than that small region. So why do we need all these bacteria isolated? Once we have it in a plate, the things that we can do with that is endless. So we can study their growth curves. We also have a plate reader inside the chamber. So we have actually we have double koi chambers, which means 4 people can use at the same time. So in one of the chambers we keep a plate reader, the liquid handling robot. So we make a huge dilution and then put it into the plate reader and do the growth curves. If we inoculate minimal media at this point and introduce different oligosaccharides, anything, amino acids, anything we need, we can also test the nutritional physiology of these bacteria. And we can test several plates at the same time because we have plate stackers, and once we can load about 20 plates at a time and it will keep giving us, spitting out data. The problem is to analyze all these data and to understand what it means. We can also, since we know what each well has, we can also build up communities. So depending on what we have and their nutritional requirements and their growth phases, we can pick certain wells and build our own communities, and we can colonize mice with them and see what happens. The in vitro system that I showed you before, the SHIME system, you can use that to do that too. So if you know your communities, you can add the community instead of adding the whole fecal sample. Adding the whole fecal sample means you are adding all the bacteria, all the fungi, protozoans, and everything. But at this point, it's more cleaner, although it's not the most natural way to do the experiment. But if you do not build your own communities, it becomes very difficult to do any kind of experiments. The other thing, the way to do it is if you give a fecal sample that has all these bacteria, if you see a phenotype, then you can go back, make culture collections from the mouse fecal sample and study them and understand what bacteria the mouse was able to get. Finally, if you are satisfied with your library, What we have to do is we have to pick every well again, grow them in regular tubes, and make stocks and freeze them again. So at this point, we have our bacteria in the arrayed library that we can work with, and we also have each bacterium in a glycerol stock in the freezer. So if there would be any failure in culturing, keeping them, we can always go back and pull it out from the freezer, from the vials. The crimp vials are actually better to hold them for longer periods of time because there's no oxygen getting into the vials because they're tight. But it's very difficult to keep these copies as they are in 96 well plates because we can only put an aluminum foil, seal them, and put them in the freezer. There is no way to keep them anaerobic, frozen for a long time. Finally, I will like to talk to you a little bit about probiotics because probiotics have made a great influence on our eating habits now, and it's filling up shelves in the supermarket. So what are probiotics? Probiotics is the name given to a group of bacteria that actually is believed to enhance the health of human beings. Prebiotics are the food that naturally enhances the growth of probiotics. The probiotics that you use, that you buy from the store, keep in mind are just the lactic acid digesting bacteria. They are not human isolates. Maybe they do something in your system, but I'm not very sure. I prefer to get the prebiotics into your system so that you can boost naturally your own bacteria. The word probiotics goes a long way. In the 1800s, Elie Mishenikov, maybe you've heard of him, he won the Nobel Prize for discovering the phagocytosis, believed that our stomach carries good bacteria that help us in good health and live longer. At the same time, he observed that Bulgarian pheasants ate sour milk every day and they lived for a long, long time. Maybe he didn't realize that they were also living a stress-free life, which we don't have today. So then he said that if you eat sour milk every day, you can live longer, healthier life. With that, the billion-dollar yogurt industry was born, which he didn't realize at that time. I'm not saying that you should not eat yogurt, but just keep in mind that it contains lactic acid bacteria and just 8 or 10 different bacterial species. It's much, much better to eat the prebiotics so that you can enhance your good bacteria. You have any questions at this point, or can I just, it's like the... Okay, okay. This is Dr. Jeffrey I. Gordon in the center. His vision and his leadership coined the word microbiome and the microbiota. And it also told us that we cannot blame our parents for the genes that we got from them for our health, and you are in charge of your microbiome. So you can change it. You can lead a healthy life. And so it's up to us to feed and keep your bacteria healthy, the population. And don't forget, you don't think you are ever eating by yourself, even if there is no one sitting at the table. You have millions of friendly bacteria eat together with you. Thank you.